So the idea on all of this is, how do we study the genome? How do we study this DNA and RNA stuff? So the first thing we're going to get into is how to clone something. When we clone DNA here, we're not cloning in the sense of your X-Files idea. Okay? We're taking a small piece of DNA and making a copy of it and putting it in something else. So for example, there's lots of uses for this that you might be aware of or might not be aware of. Okay, so I'm going to say we have a cell here. Okay? And we want to pull some DNA out of this cell and put into another cell. For example, we might want to take we'll talk about this with GMOs here, we might want to take a gene from a bacterium called Bacillus thuringiensis, which can kill insects, and put that into a plant. A plant would then kill the bacteria or pathogens. So there's lots of uses for this. What if we want to take a human cell and take out what makes insulin? And instead of getting insulin from things like pigs, we can put that gene for insulin into a bacterium and let that make insulin for us. So we've done that also. So there's some real uses to all of this. So let's start with, before we start talking about this process, describing what a restriction enzyme is and what recombinant DNA is. All right, so a restriction enzyme, we also call it an endonuclease. Endonuclease. The term endo implies what? It's the inside, not the end, the inside. Think of exo. If you got an exoskeleton, it's on the outside. You have an endosskeleton, it's on the inside. Nuclease, the ASE means it's an enzyme, and the nuclea means it's DNA, or nucleic acid, I should say. So this is an enzyme that works in the middle, or targets the middle of DNA. That is an endonuclease, and here we call it a restriction enzyme. Now there's lots of different restriction enzyme types out there, but we're just going to talk about one type of them. So if you continue in biology, there's a lot more to this than I'm letting on. All right. So let's talk about bacteria and pathogens. All right, you get sick, right? What happens when you get sick? You go out, someone coughs on you, you get the flu virus. What do? You have an immune system that addresses that, right? Okay. Well, bacteria are the same way. Here's a bacterium. All right, let me get an alternative color here. And we're talking about bacterial viruses. We call them bacteriophages. Here's a phage. Here's the bacterial DNA. And the phage will put its nucleic acid inside of our bacterium, possibly taking over the bacterium and ultimately destroying it. Okay, so bacteria can get sick just like humans can. We have an immune system, don't we? Bacteria have a kind of an immune system, if you'll think about it that way. They have these restriction enzymes, and I'm going to put RE for restriction enzyme, and they have restriction enzymes that will chop up this foreign DNA. It's an endonuclease. It goes in, it's like a pair of scissors, and it chops up the DNA that has been entered in from this bacteriophage. If we do that, we can protect the bacterium from a takeover. Now, the reason it does not chop up its own DNA is because it's modified with methyl groups. We're not going to get into that, but someone asked that question on another class. So anyhow, bacteria have these enzymes that will break up foreign DNA. Okay? So that's kind of handy, isn't it? Well, another handy thing about restriction enzymes is they can detect, or we're going to say recognize, specific sequences. They are what we call site-specific. At least a class of restriction enzymes is site-specific. So when it detects a particular sequence, it can go into that sequence and cut it up. All right? So for example, let's say we have a sequence that is A, 5 prime, A, T, G, A, T, C, G, T, A, T, C, G. And the comparable one would be what? T, A, G, T, A, G, C, A, T, G, C, right? That's just base pairing rules. What if we had a restriction enzyme that could come and everywhere it sees this sequence, it comes in and like a pair of molecular scissors, if you will, comes in and cuts that DNA. So right where we have this, we call it a cut site or a recognition site, it cuts that DNA. So when it separates, we've got A, T, T, G, A, and then right the other here, A, G, T, T. But we have it, it's not a cut like a pair of scissors straight down, it more cuts diagonal. So here we're going to have an A, G, C. On the other end of the DNA, we have a T, C, G, T, C, G, A, G, C. Bless you. So now we have two pieces of DNA here, don't we? Before we had one piece. Yes? We're here?
No, over the fourth one.
Yes. Good catch. Good catch. All right. So now we have separated our DNA into two pieces. Now imagine this happening multiple times on a piece of DNA. All right. Now, this is at a specific site. So everywhere we have a GAATTC, this restriction enzyme will come in and like a pair of molecular scissors cut our DNA. Now we have a special name for these ends. We call them sticky ends. Okay. You guys ever play with Legos? You know, and one Lego on the top has the bubble parts and the bottom has the holes. Well, we're going to try to basically treat these like Legos. Okay? Now let's talk about cloning. So this is restriction enzymes. Restriction enzymes can, not always, leave these sticky ends. Okay? So I've introduced you to restriction enzymes. Now let's get into what this cloning is all about. And we'll come back to these here pretty soon. So let's say we have our cell here, and we have all this DNA inside of our cell, and we want a piece of that DNA. So we take that DNA and we pull it out. And we chop it, we cut it with our restriction enzymes. It is going to leave an overhang, a sticky end on each of the ends. Let's just say, for instance, that restriction enzyme site was GGGCCC, and on the other it would be CCCGGG. And let's say right here we have a GC overhang. Over here we would have a C and G. Does that make sense? So we've cut our DNA, right? We've specifically looked for a target sequence and we've cut it, and we have these sticky ends, which are like the ends of Lego blocks. All right? Now what would happen? What's kind of neat about Lego blocks? You can take one Lego block and put it on another one, right? Okay? Well, let's say we want to get one of these pieces of DNA into another cell. So what we're going to do is we're going to take something called a vector. Okay. Now this vector is also, you call this circular DNA is a plasmid in this regard, but we're going to call this a vector. And here's actually a picture of one of those vectors. And do you see how we have this little red arrow here? And do you see how we have a bunch of these abbreviations? NOT1, SAC1, SAL1, ECOR1. You see that? Okay, that's what we call a multiple cloning site. Each one of those abbreviations is a specific restriction enzyme. So let's say we cut this vector with the same restriction enzyme as we cut this DNA with. What would we have here? Let's say we cut it here. What kind of overhang would I have? A GC, and what would be over here? A C and a G, right? So what would happen? Let's further suppose I took this cut vector and I mixed it with this cut DNA from this cell. Think of Legos. This piece of DNA would insert here. We call it an insert for that very reason. And we would make something like this.
So the DNA would just fill in the cut?
Correct. Correct. We call this a vector and this an insert. This whole thing is referred to as recombinant DNA. Okay, let me show you the definition there. Right here, recombinant DNA. So we've taken DNA from one and combined it with DNA from another. Fantastic. Now let's just further suppose this has some type of a gene on it that we are really curious in. It could be a gene for an insecticidal protein from Bacillus thuringiensis, for example. So let's take this, and genes code for what? Ultimately into the P. Protein, right? We're coding for a protein with this gene. So we're going to take, not drawn to scale here, we're going to take a common bacterium, E. coli. This is probably the most transformed bacterium out there. You can transform other ones, but usually E. coli is the one you use. And you use agrobacterium if you want to, anyhow. You're going to take this and you're going to transform E. coli and put your plasmid in there with your insert. And basically ask the bacterium, why don't it express what is on that protein? Excuse me, what is on that foreign DNA I put in you? Okay? This is cloning DNA right here. This is the idea of cloning something. Here's a picture of it from your book. Here we have cloned DNA. So looking at A on here, what do? We take our DNA and we cut it with those enzymatic scissors, the restriction enzymes. It leaves these sticky ends. I've shown with G and C here. B, we cut our plasmid, our vector, with the same restriction enzyme, the same molecular scissors. Similarly, it leaves the same bases on our plasmid that are exposed, the same sticky end. Then we take them and we combine them together. We make a recombinant piece of DNA. We then take this and put it into a bacterium like E. coli. This whole thing is cloning DNA. Then we ask the bacterium, make this protein for us. Yes, you can make a lot of protein this way. Now, we'll get to that with complementary DNA later on, but for example, it used to be insulin. You would get insulin from pigs. Okay? There are some downsides to that. There's actually a lot of downsides to that. So now what we do is, and it's not exactly this straightforward. There's more to it than this. But basically a similar analogous process than this, we've taken the gene for insulin, we've put it in bacteria, and we have the bacteria make the insulin. Correct. I mean, we can take a gene for insulin from a human, typical insulin gene, and put it in some other organism, and they make a lot of the insulin for us, instead of us having to get it from a pig. All right. Another one is that, and we'll talk about Bacillus thuringiensis here with genetically modified organisms. So this whole thing is cloning DNA. We take it from another source, combine it with a plasmid here, and then we put it into typically a bacterium. Now there's a lot more to it than I'm letting on here. So, there's a lot more here. All right. Now there's a problem here. Let's say, remember this is a test question. This was a test question that introns are found in eukaryotic organisms, right? But introns are not found in bacteria, right? We're not going to be able to pull those introns out and recombine the exons. Remember that part? So what would happen if we had some type of a gene with introns in it? Could we take just a gene with introns and exons and do this procedure? No. Because we'd be putting introns and exons into a bacterial background, the bacterium would be like, what do you mean do with it? I don't know. Well, it makes something, but not what we want. So we got to kind of do a little clever trick here. And that's where we get to this cDNA, or complementary DNA cloning. So let's go back, and obviously this should be ingrained in your brain right now. DNA leads to RNA, leads to protein, right? Here is our DNA. We have a series of introns intermixed with exons, right? And I'm going to say the orange are our exons and the white are our introns. Right? Okay. When we make a copy in RNA, we're going to copy both the introns and the exons. Now it'll be in a slightly different format, one being made of RNA, one being made of DNA, but we've copied our introns and our exons. But before we go to protein, what do we have to do to this? We have to remove the introns. And then we splice back together the exons. So if we did like the strategy before, we took the genomic DNA from a eukaryotic organism, we would also get the introns, wouldn't we? That's the problem. We want the protein that this thing makes right here. Or we want to look at the sequence. Maybe not want the protein. We just want to see possibly how the introns were taken out and which specific exons were kept. So we might not even want the protein specifically. We just want to see what exons were combined back together. Okay? So what we're going to do is we're going to take the RNA. Here on the diagram is that brown strand on the very top. We're going to take the processed RNA, and we're going to get another color here. We're going to make a DNA complement to our RNA. And we use something called reverse transcriptase. Okay, so we're going to make a DNA complement to our RNA. Then through a series of enzymatic reactions, we are going to then make a double-stranded DNA. Not of the gene, but of the processed RNA. So this has of what? What does this have in it? Just exons. Right? This had exon 1, exon 2, exon 3. Intron 1, intron 2, intron 3. The RNA had exon 1, 2, and 3, and the intervening introns. We let the cell take them out. Then we spliced together our exons. We then made a DNA complement of it through the enzyme reverse transcriptase. And through several enzymatic reactions, we come out with a double-stranded piece of DNA that is just of exons.
And that would be protein.
No, this would be put into, say, a bacterium and then made into a protein. Let me erase that protein there. So this would then be put into something like E. coli. And that E. coli would take this and make it into protein. Now, sometimes you don't want the protein. Maybe you just want to see which exons came together. That's also important sometimes. So you could actually do more than that. So you could say, did exon 1, 2, and 5 come together? Or did exon 2, 3, 5, 7, and 9? Which exons were that product? Or here we just might want to make it into a protein. Okay, that's cDNA cloning. All right. Questions on that? Alrighty. Okay, we talked about PCR a lot last time, right? Polymerase chain reaction, Curry Mullins, 1993, chemistry, born in Lenoir, North Carolina, kind of close to where my dad's from. Anyhow. DNA sequencing. Now this is dye termination sequencing. There's other types of sequencing out there. This is one that we do here. There's next generation sequencing. There's 454. There's a lot of different sequencing techniques out there. And this is one way we can do it. All right. So throughout this, oh, phone's not broken. It's fine. All right. So throughout the semester, I've drawn a bunch of letters out here, right? That's a sequence of bases. Okay. So how do we determine a sequence of DNA? All right. Let's start with a very basic sequence here. Let's say GATTA. Okay? This is a very short sequence of DNA. And before we start looking at this sequence, let's go back. Oh, bless you. Bless you. Let's go back and look at that whole DNA model. Okay? When we had DNA and RNA, we said that RNA had an oxygen hydrogen here, right? When we removed that oxygen, what did we make? We made deoxyribonucleic acid, known as DNA. Okay, this is a 3', 2', 1', 4, and 5'. All right. So, fine, great. Now, here we're going to use a modified nucleotide. Here, instead of having an OH, we're going to have a hydrogen. We're also going to have a specially labeled base, a color tag, if you will, on the nucleotide. Okay, now, because it's here, we call this a dideoxy. Di meaning 2 and deoxy meaning it's missing those oxygens. And I want to tell you, when this special base integrates into the strand, it's going to stop synthesis because this hydrogen can't connect to another nucleotide. So if this thing integrates into our strand as it grows here, it stops. Termination happens. No more bases added. That becomes important later on. So when this adds to our strand up here, termination happens, no more bases added. Okay? So, let's say round number 1 of PCR. We add a C, and a T, an A, and this fourth one, we added one of those modified A's. Can we add another base after this? No. It's terminated the product. So this product size is C, T, A, and a labeled A. Okay? Do another round of sequencing reaction. We do this in a thermocycler, what we use to do PCR with. This one, a G, a T, and an A here. Can we add any more bases here? No. So now we have G, T, and a labeled A. Okay? Not to be confused with 5. Yeah, someone got it. All right. Here, we label a a C, just a single labeled C. Because it has that 3' hydrogen, can we add anything past this? No. So we have a label C. All right. Another round. A C, and a T. Anything you add past here? No. C, T. Finally, we get the full Gamillicuddy. We get the C, the T, the A, and a labeled T. Okay. So we now have a C, T, A, and a labeled T. All right. Now, have you guys had your Who's the Daddy lab? Who is thy father lab? Or they name it now. All right. Well, do you remember if you've had your lab? Have you guys had the lab? No. Okay. I'll tell you a rule here. We can size DNA. We're gonna pull it through a gel. Sometimes that gel is agarose. When you do your lab, you'll have a tank about like that big, and you have a little sheet of white material in there. It's like Jello. Okay, you can move DNA through that agarose medium. But you can also move it through something called polyacrylamide, basically plastic, or plastic-like material. And when we move DNA through this, it's going to move from the smallest size to the largest size. Okay, we call that movement migration. All right, so now I want you to imagine this tube. It's a very thin tube, call it a capillary. And in this tube, we have a window. I'm going to make this tube a little bit bigger, actually. Okay. We have a little window here, if you will. Okay. And here we have basically a camera and a source of... Okay. So we have a way to detect what moves past here. And this goes to a computer. This is a rough rendition. We have one of these in McGee Hall. I use it. Well, I have used it in the past a lot. Okay, $100,000 plus. It's an expensive piece of equipment. All right. So now we're going to have a positive charge down here. DNA is negative, so it's going to be drawn to the positive. And if it moves based on size, what base will move here for the first? The base of these sizes. The C. So all these bases are moving, but that C will move through here the first time. And our camera connected to the computer will plot this out. It has a time and signal thing here. First thing will be a spike up, and that spike will correlate to the C, which that labeled tag correlates to. Okay? That will pass through, and what will be next in the queue? A, C, T. What will the computer register here? A, not a CT, but a labeled T. It's not going to see the C, it's just going to see a labeled T. Okay? Then the electrical pulse goes a little bit more. CTA. So we'll have a CTA. It will only detect the labeled A. It won't detect the C or the T. It's only going to detect that label. We'll get a spike up. A. Not GTA, and CTA. Sorry about that. All right. And we continue this whole process, and pretty soon we're going to get another spike for the A, and we'll get a final spike for the T. We're not determining any of these bases. We're only determining the signal of that base at the end that terminated the reaction.
Why aren't you paying attention to any of the other?
Because they're not labeled. You determine what these are by inference, by size. Because if this moved the last, and the one that moved before it had a labeled A, then your assumption is that what is here is A. You can't, when you detect this one passing through, you don't know anything about these. You just know the one that came before it had a labeled A. So by just your assumption, that has an A there. And it comes off really fast on the instrument. I mean, you'll see it happen like 2 or 3 times a second. You'll be watching it have a spike, and a spike, and a spike, and a spike, and a spike.
When we did the lab, we did it with just a bunch of different dyes. It actually took a minute, though. It was really slow.
Okay, so different. So that's a gel electrophoresis. It can take 30 minutes, 45 minutes. But this can also take about 70 minutes. It takes a while. So you're asking very small particles to move through a medium, so it does take some time. But we're talking about, okay, so if I started from here to here moving, it's going to take a while. But what I'm saying is this piece, and then a few seconds later, this piece would move. Then this piece right here. So you're getting signals pretty fast, but the DNA itself is moving really slow. I guess the best analogy is think of a train going from here to Pittsburgh. Okay? Really, that train's going to take a while to go from here to Pittsburgh, right? But if you're standing at a train track, watching the train go by, it's going by pretty fast, each car. So that may be the best analogy. That's actually a pretty good analogy. That from the perspective of the computer, the train tracks, or the train's going, but it takes a long time for the train to go from Alabama to Pittsburgh. Okay. Now, there are other next generation sequencing techniques. One's called 454. There's all sorts of them. And the reason I like to mention this is because there is a pretty good chance that you or your children will have your DNA entirely sequenced at some point. Okay, now, it used to be, okay, this, when we run our system, we can get about 500 base reads per reaction. We can do 8 reactions at a time. So a couple thousand base reads. It used to be people that had to do the sequencing would take days and then have these long slab gels, literally like almost the length of this table of glass, and use radioactively labeled things. And you could read maybe 50, 100 bases. 200. It varied about how long the gel was. You have to have radioactive things, and it was not very pleasant. Okay? But the point I'm trying to get here, this little axis here, time, right, as we go forward in time, and your cost per base. Before, there was a significant cost per base and time and effort. And what we're getting is pretty much a thing like this. Okay? 15, 20 years ago, we were here. 10 years ago, we're here. We are getting a progression to the point where it's reasonable to take an entire human genome and have it sequenced. Now, I'm not saying it's going to be like in Gattaca or anything like that where you take a hair and then like pops out, here's the sequence. And we haven't gotten there yet. But if you follow the extrapolation, if you follow the trend line, it could get there. Well, the trend line will get there at some point. So, foreseeable future, I don't think it's going to happen in the next few years, but down the road in your lifetime or your children's lifetime, possibly could be when you go to the doctor and have your blood drawn, it's also sequenced. The cost is getting there. Okay, the time it takes to do it is getting there. And how it does it is it massively paralyzed. It's a massively parallel system. So you're having all of these reactions run at once. Tens of thousands of multiple reactions run one on top of each other. And overall, it makes your sequence. We're not going to get into next generation sequencing in here, though. But something to think about. Why is it important? No. Well, no. Yeah. Why would having your whole genome sequence be important? What if you're looking for a particular gene for cancer or some ailment? What if your treatment is determined based on what sequence you have? So a treatment that is good for one type of genetic sequence might be not so good for another type. So you can actually modify your treatment such that it suits your genome. Already being done. Okay, this happened. I was teaching a genetics lab a couple of years ago, and people, part of it is you take the DNA and you swish it around your mouth and you spit it out in a cup, and they take and you purify the DNA. You're looking for an Alu insert, but neither here nor there. Anyhow, and everyone gave me their DNA at the end, and no one asked, "What are you going to do with it?" To me, if you get something like this where cost is starting to go down, I want to know what you're going to do with my DNA. What's another thing? A fear is, what if you're prone to a particular cancer and you go in for insurance? Possibly. Okay, just theoretically speaking here. And they say, well, based here, you have an 85% chance of getting a cancer here. Well, I don't know about that coverage now. So. Now that, will that happen? Who's to say? Okay, but, you'd ask the question, why is it important? It is important. Okay. Or if you ever seen the movie Gattaca? What if part of after a date, you think you might be interested in someone, right? You take part of their DNA, and it's like, well, this person has these, and these characteristics. So, just something to think about. All right, one last thing here, and we will be done with this material. GMOs, genetically modified organisms. Now, remember we talked about taking some type of a protein from an organism, right? Or what could you do that cloning for? Okay, there's a lot more that I'm not letting on here, so I'm just trying to fit this in the time we have. But let's say we had a bacterium. And this bacterium, as part of its life cycle, released a protein. And this protein could kill an insect, kill a bug. All right, so here's a little bug. And this protein, when ingested by the insect, could kill that insect. It's insecticidal. There should be a benefit because the bacterium would basically have a medium. Growth medium to grow in. Okay? That's fantastic, but what's the problem here? Or what's the advantage here? What's the advantage here?
It'll be able to grow without being eaten.
Right. Or it will take the dead insect and use that as as food, basically, make more of itself. But what could you use that for? What if we did this? What if we took the gene that codes for this protein, theoretically, and what if we put that gene into a plant? And now what if this plant also made that same protein that killed this bug? Couldn't that help protect the plant? Couldn't we just do that and not have to spray pesticides on plants and protect plants that then make the protein? Well, we've done this for decades. We call this bacterium Bacillus thuringiensis. The protein here is called a cry protein. The gene, lowercase c, is a cry gene. Okay? And we can take these cry genes and put them into plants. Has anyone ever heard of something like, you see this in the media occasionally, BT corn? Has anyone ever seen something like that? No? If you do a Google News search for it, you'll find something about it. The BT refers to Bacillus thuringiensis, the bacterium from which we get the cry protein. Okay. So what we've done is we've taken this gene and go back to that idea of cloning, right? We've taken it and basically we've put it into here. Okay, it's a little bit more involved than what I'm letting on here, but basically put this into this. This then makes a cry protein and it kills insects. Okay, so again, you'll hear about these in the media, and I'm not going to get into the whole thing with what it can possibly cause. That's another discussion. But the point here is that we genetically modify organisms all the time. All right, but it is an interesting point to think about. All right. With that, we are done with that test material. Let's talk about any questions. Would it be good if I started with the practice test? Okay, let me turn my recorder off here.